The overall goal is to monitor the course of oral Listeria Monocytogenes infection using a mouse model that more closely mimics human disease than conventional intragastric or intravenous models. The first step is to contaminate a piece of bread with Listeria. Next, mice are fed the bread to induce a natural foodborne infection. At various time points post-infection, tissues are harvested for analysis. Finally, the total number of Listeria present in each infected tissue is determined by plating on selective agar media. Using this model, BALB/c mice are highly susceptible to infection, while C57 Black 6 mice clear the infection rapidly.
My former graduate student, Elsa Ghanem, developed this novel model of oral infection after we discovered that intragastric inoculation of mice resulted in almost immediate systemic spread of Listeria monocytogenes. This complicated our efforts to identify the pathways used by the bacteria to translocate across the gut mucosa. The main advantages of this foodborne model are that it is not physically invasive or traumatic for the mice, and there's a reproducible delay of 36 to 48 hours before the bacteria disseminate from the intestines to the peripheral tissues. Demonstrating the procedure will be Tanya Myers-Morales, a research scientist, and Grant Jones, a graduate student from my laboratory.
Begin by cutting white bread into small cubes, using a sterile scalpel blade and avoiding the crusts. Place individual pieces into